What is this?
Leaf spot.
Eh? What is this?
Who said lemon?
What is this? Eh? What is with what?
With what?
With spot. What type of spot is that? Anyway, this is our plantation. That's a cashew plantation. It belongs to UNEP, Agric faculty. Are you okay? Please. So now, this is a cashew plantation that belongs to the university, okay? For practical sake. What is the name or what is the scientific name for cashew? The scientific name for cashew is what? Anacardium what? Occidentale. Are you okay? Please. Anacardium occidentale. That's the scientific name of what? Cashew. Are you okay? So, and my specialization is pathology. So, in pathology, you know, it deals with what? Disease. Eh? It deals with what? Plants what? Diseases, isn't it? Yes. And what is plant disease? Abnormal or what? Dysfunction of a plant, isn't it? As a result of what? As a result of what? Biotic or what? Abiotic factors. Are you okay? As a result of what? Biotic and abiotic factors. So abiotic causes are what? Please. So I said that what? They are caused by what? Biotic as well as what? Abiotic factors. And what are abiotic what causes of disease? Eh? They are caused by what? Non-living organisms. And biotic aspect, what are they? What are the living organisms that normally cause disease, plant diseases? What? Fungi, bacteria, nematode, viruses, isn't it? They cause what? Please. Biotic. They cause what? Plant diseases, isn't it? Yes. So you can see a what? This is a leaf, isn't it? Yes. And it's attacked by what? A pathogen. Are you okay? So what is the name of that pathogen? So this one we call it red rust. We call it what? Red rust. And it's caused by a pathogen called what? Algae. Cephalarius. We call it what? Cephalarius species. Are you okay? Cephalarius what? Species. Here you can see at the back, you can see some what? So symptoms there. Are you okay? So this one we call them what? Anthracnose. Are you okay? Anthracnose disease, and it's caused by what? A fungi called what? Coleotetrachum glosporoides. Are you okay? It's caused by what? Coleotetrachum glosporoides. Are you okay? It's caused by Coleotetrachum what? Glosporoides. Are you okay? It's a fungal pathogen. Are you okay? That causes what? Anthracnose disease on the what? On the plant. Are you okay? So we have what? Several other pathogens. Are you okay? So we have others. We have what causes what? Gumosis. And gumosis. Have you heard of gumosis? Yes. You see exudation, something like a chinga-like. Have you ever seen a tree? You see like a chinga-like or some fluid oozing out. Are you okay? So we call it what? Gumosis. Are you okay? So and it's caused by what? Lasidioplodia theobromine. Are you okay? So it's a pathogen that what causes what? Gumosis in what? On what? A plant. Are you okay? Do you have lab? No. Do you have lab? No. Main campus. Main campus. How many do you have lab? No. So now, our reason for being here, we apply for what? Equipment. Are you okay? You know, all along we'll be teaching you what? Theory, isn't it? Yes. So your voice. All along we'll be teaching what? Theory. So we have to add what? Practical. Are you okay? Yes. So we've applied for equipment from an organization called what? Seeding Lab, which is in the US. Are you okay? So, and with their agreement, they will be supplying us 20-footer container worth equipment. Are you okay? So clap for the Seeding Lab. Yeah. Yes. So when this equipment arrive, we'll be able to do what? Our practicals in the lab, isn't it? Yes. So in the lab, what we normally do, like to be able to, this what? A spot, a blight, isn't it? And it's advancing. Are you okay? So in order to be able to know the causal agent of this, what do we do? We do what we call what? Isolation. Are you okay? And isolation, we do it in the lab under aseptic what? Condition, isn't it? So what we normally do in the lab, is you take this is a disease plant, isn't it? So you take your knife or razor blade, then cut the disease portion, isn't it? Together with the adjacent healthy portion. Are you okay? Then you sterilize it in 70% ethanol.
Okay.
70% what? Ethanol. So after that, you wash it in sterile distilled water or double distilled water. Are you okay? So Incident Lab supply us with these autoclaves and other lab equipment. We'll be able to go to the lab. We'll do the what? Autoclaving. We'll prepare our media. Then we'll be able to what? To capture them. So if you what? After the disinfection and blotting dry on the white man's paper, then with the aid of your forceps, you pick it and put it on what? On the protective what? Destrous what? Media. Are you okay? The agar. So that's food for them. Microbes eat them. Are you okay? So after that, you parafilm it and what? And put it in an oven. Are you okay? Or incubator. At the temperature of what? Between 25 and what? 28. Are you okay? So you leave it for 3 to 7 days. After each what? It will start what? Growing, isn't it? After growth, then you do what you call what? Microscopy. Do you have microscope?
No.
Right? I want to hear from them. Do you have microscope? No. You said they don't have microscope. So we are waiting for the seeding lab to what? To provide us with what? Microscope. Are you okay? So once they what? Once they supply us with what? The microscope, you'll be able to what? View or examine this pathogen under what? Microscope. Are you okay? After the growth in the petri dish, they produce what we call what? Mycelium. Any question from students?
No question.
Are you sure that there's no question?
Yes, sir.
Yes. So I said we are waiting for equipment from which organization?
Seeding Lab.
Seeding Lab. Are you okay? So once we get the equipment, we'll be able to do what? The practicals here instead of going to what? Sunyani. Are you okay? Yes. So the topic for today is what? From the field to the lab, isn't it? Where is the lab?
No lab.
Where is the lab? No lab. Where is the lab? No lab. Where is the lab?
Seeding Lab.
Where is the lab?
Seeding Lab.
Yes, so we are done for today. So that's the pathological aspect of it. Now we'll move to the other class. Then we'll continue with the what? We'll continue with the what? The agronomy aspect of it. It's here to take us. Yes.
Demonstration farm of School of Agriculture and Technology. Is that okay? University of Energy and Natural Resources. We have several plants here. We have cashew trees, we have cassava plants, we have maize plants. Is that okay? As we speak, plans are underway to conduct some trials with IFDC, that is International Fertilizer Development Center. So by the time you resume from holidays, we will have begun the field trials. Now, basically, before somebody goes into crop production, there are 4 main activities which should be considered. We have pre-planting activities, planting activities, post-planting activities, then harvesting and marketing activities. So the farmer has to consider all these activities before going into production. Now, when we talk about pre-planting activities, we're talking about those various works or activities which are performed before actual planting, right? So we talk about activities such as selection of a parcel of land, selection of crop varieties. Is that okay? Yeah. The kind of crop to grow. Then we talk about land preparation, that is site selection. Are we together? So under land preparation, we have land clearing and soil preparation, what we call soil tillage. Are we together? Yes. Now let's take site selection. Assuming you want to go into cassava production, what are some of the factors which will determine the type of land to choose? I'm asking you. One of them is the nature of the land. So you look at the fertility of the soil. Is that okay? So basically, the soil of the area should be very fertile. It should contain sufficient amount of plant nutrients, nitrogen, phosphorus, potassium, calcium, what have you. Are we together? Yes. Good. The soil of the area should be free from toxins. It shouldn't be toxic. If it is toxic, it will be harmful to the crops. So it should be free from toxins. Are we together? Yes. Then it should be free from harmful pathogens. like viruses, bacteria, fungi, protozoa, then parasites. In other words, metazoa, scientifically. Altogether, metazoa are parasites. So you make sure the soil of the area is also free from harmful organisms. Altogether.
Yeah.
Good. Then the soil should also be very rich in organic matter. The soil of the area should be rich in organic matter. You also have to consider availability of labor. You can't do everything alone. You need to fall on others. Are you getting it? Yes. So human resources should be available so that you get them for land clearing, soil preparation, planting, then to execute cultural practices like weeding, fertilizer application, thinning out, breaking out, together, roguing, and whatever you can say, control of pests and diseases, and even pruning, harvesting, depending on the kind of crop under cultivation. So labor is very, very important. Are we together? Yeah. Then you also have to consider the funds you have, the financial status of the farmer. Ask yourself whether you have enough funds to start a business, the family business. Because whatever you do depends on money. Please, am I making sense to you?
Yes.
Are you very sure? Yes. Sure? Yes. All right. We can mention all the factors. Yeah. Later you look for some. Yes. Now, these are some of the pre-planting activities. The third one is soil preparation. So land preparation, we have soil, land clearing, and soil preparation. We have several methods of clearing our farmlands. Yes. We have the use of fire, where farmers will set their farmlands ablaze just to get it cleared. Are we together? Yes. Please keep quiet. We have the use of simple hand tools, where we use or we make do with cutlasses, hoes, and axes. Is that okay? Yeah. Then we have mechanized system or method, where we make do with bulldozers, tractors, and what have you. We use weedicides to clear weeds. We use arboricides to clear trees and woody shrubs. You learned it in morphology, right? Very good. So you use arboricides to clear all those ones. Then the fifth method is the use of animal-drawn implements. When you go to north or northern part of Ghana, they mount plows and harrows on donkeys. Animals like donkeys, bulls, are we together? Yes, for cultivation purposes. So basically, these are the 5 main ways of what? Preparing our farmlands. Are we together? Yes. Now, after you have prepared your farmland, I mentioned tillage. Plowing, raising beds, ridging, mounding are all tillage practices. So after you have prepared the land, the next is what? Plant. I believe by the time you come, we may have established a lot of field trials, and we will come to the field with you. I wish you a very safe journey.
Thank you.
Our warm greetings to your family. Please, please, and please behave and behave well. Don't throw away your notebooks. Continue to read them. Yes. Are we together? Yes. And live exemplary lives. Yes. Thanks so much and bye-bye.